Hello and welcome to Biology Explained. Today we are going to be exploring an important topic endocytosis and exocytosis. Imagine a cell as a busy city, alive, controlled, and constantly active. Not just any city, but one with strict border control where nothing enters or leaves without permission. Nutrients, hormones, waste products, all must pass through a highly regulated system. But here's the challenge. Some substances are simply too large to pass through the cell membrane by diffusion or even facilitated transport. So, what does the cell do? It wraps them up and moves them in bulk. This is where today's focus comes in endocytosis and exocytosis. These are not just transport processes, they are life sustaining mechanisms that allow cells to feed, communicate, defend, and maintain internal balance. By the end of this, you won't just understand what they are, you will understand why they are essential to survive. Let's begin with the foundation. Endocytosis simply means bringing substances into the cell using vesicles. Break down the word. Endo means inside, cytosis refers to the cell. So, literally, the cell bringing something inside. But the most important keyword here is vesicle. A vesicle is a small membrane bound sac, like a tiny bubble formed from the cell membrane itself. During endocytosis, the cell membrane folds inward, surrounds the material, and then pinches off, forming a vesicle inside the cell. This is not a passive process, it requires energy. That means it is a form of active transport. Now consider the opposite process, exocytosis. This is the movement of substances out of the cell using vesicles. Exo means outside, and the mechanism is just as fascinating. A vesicle inside the cell moves towards the membrane, fuses with it, and releases its contents outside. This is how cells secrete hormones, release enzymes, and expel waste. Again, energy is required. Both of these processes fall under a broader concept known as bulk transport, the movement of large quantities or large particles across the membrane. Unlike diffusion or osmosis, these processes do not rely solely on concentration gradients. They involve physical restructuring of the membrane itself. If a substance is too large to pass through channels or carriers, the cell uses vesicles. Simple, efficient, and powerful. At the core of all this is energy. Anytime you see the membrane bending, vesicles forming, or membranes fusing, you should immediately think of ATP being used. These are active, controlled, and highly coordinated events. A simple way to log this into memory is this endocytosis is engulfing, bringing things in, while exocytosis is exiting, sending things out. Once that sticks, everything else builds naturally from it. Now that the foundation is clear, let's go deeper into how cells actually perform endocytosis. Because not all cell eating is the same. Cells don't randomly engulf materials, they do it in a specialized way, depending on what is being taken in, how large it is, and whether it is needed specifically. There are three major types of endocytosis, and each reveals how intelligent and selective cells are. First, phagocytosis, often described as cell eating. This is the process by which a cell engulfs large solid particles, and the keyword here is large. We're talking about bacteria, dead cells, cellular debris. This is not subtle, it is aggressive and purposeful. Instead of forming a small vesicle immediately, the cell extends part of its membrane, called pseudopodia, literally false fits. These extensions reach out, wrap around the particle, and enclose it completely, forming a large vesicle known as a phagosome. But it doesn't stop there. The phagosome fuses with a lysosome, an organelle packed with digestive enzymes, forming what is called the phagolysosome. Inside this structure, the engulfed material is broken down, useful components are absorbed, and waste is eventually expelled. This process is critical to immune defense. Specialized cells like macrophages and neutrophils use phagocytosis to destroy invading pathogens. When bacteria enters the body, these cells detect, engulf, and digest them. Without this mechanism, even minor infections could escalate into serious threats. Phagocytosis is not just feeding, it is survival. Next is pinocytosis, often referred to as cell drinking. Unlike phagocytosis, this process involves the uptake of liquids and dissolved substances. The key concept here is that it is not specific. The cell is not targeting a particular molecule. Instead, it simply takes in extracellular fluid along with whatever solutes are dissolved in it. The membrane folds inward slightly, forming small vesicles that contain fluid and dissolved substances. This process occurs continuously in many cells, allowing them to sample the environment, absorb nutrients, and maintain internal balance. For example, cells lining the small intestine use pinocytosis to take in dissolved nutrients from digested food. While it may not be the primary method of absorption, it plays an important supporting role in ensuring that the cell has access to essential material. Finally, we have receptor-mediated endocytosis, the most selective and precise form of all. This is where the cell takes in specific molecules using receptor proteins embedded in its membrane. These receptors are highly specialized. They bind only to particular molecules known as ligands. When the correct molecule binds to its receptor, the membrane folds inward, forming a vesicle that brings that specific substance into the cell. This is not random, it is targeted and controlled. Often, these vesicles are coated with proteins like clathrin, which helps shape and guide their formations. This method allows the cell to absorb exactly what it needs, no more, no less. It is how cells take in critical substances like cholesterol, hormones, and vitamins. For instance, cells regulate cholesterol levels by internalizing LDL particles through receptor mediated endocytosis. However, when this process fails, such as when receptors are defective, cholesterol can accumulate in the bloodstream, contributing to cardiovascular disease. This highlights an important truth. These processes are not just biological, they are medically significant. So when you step back and compare them, the differences become clear. Phagocytosis handles large particles, cell eating. Pinocytosis deals with fluids, cell drinking. So when you step back and compare them, the differences become clear. Phagocytosis handles large particles, cell eating. Pinocytosis deals with fluids, cell drinking. Receptor mediated endocytosis is selective, targeted intake, three different strategies, one unified purpose, controlled and efficient entry into the cell. A cell is not just a receiver, it is also a sender, an exporter, and a communicator. Everything it takes in must eventually be processed, used or released. And the process that makes this outward movement possible is exocytosis. Exocytosis is the mechanism by which a cell releases substances to the outside by fusing vesicles with the cell membrane. But this is more than simple contact. It is a complete merging of membranes. The vesicle becomes part of the cell membrane, and its contents are expelled into the extracellular space. In doing so, the cell doesn't just release materials, it actually contributes to the growth and renewal of its own membrane. The process begins with vesicle formation. Substances meant for export, such as proteins, enzymes, or hormones, are packaged into vesicles, often by the Golgi apparatus. These vesicles are then transported through the cytoplasm towards the cell membrane, guided by the cytoskeleton, a network of protein fibers that acts like an internal transport system. Once the vesicle reaches its destination, it docks at the inner surface of the membrane. Specialized proteins ensure that it attaches at the correct location, preparing it for the next critical step, fusion. Using energy from ATP, the vesicle's membrane fuses seamlessly with the cell membrane. And finally, the contents are released outside the cell in a process known as secretion. Exocytosis occurs in two main forms, each defined by timing and control. The first is constitutive exocytosis, a continuous and ongoing process where substances are released as soon as they are produced. There is no waiting period. Vesicles are constantly formed, transported, and fused with the membrane. This is essential for maintaining the cell's structure as it ensures a steady supply of membrane components and proteins. Without this continuous flow, cells will struggle to maintain their integrity and function. The second type is regulated exocytosis, which is far more controlled and responsive. In this case, vesicles are formed and stored within the cell, often clustered near the membrane, waiting for a specific signal. This signal could be a hormonal cue, a nerve impulse, or a sudden increase in calcium ions within the cell. Once the signal is received, the vesicles rapidly fuse with the membrane and release their contents. This type of exocytosis is critical for communication within the body. For example, when a nerve impulse reaches the end of a neuron, vesicles containing neurotransmitters fuse with the membrane and release their contents into the synapse. These neurotransmitters then cross the tiny gap between neurons and bind to receptors on the next cell, allowing the signal to continue. Without this process, the brain will be unable to transmit information. There will be no movement, no thoughts, no coordination. A similar mechanism operates in hormone regulation. After a meal, as blood glucose levels rise, cells in the pancreas respond by releasing insulin through regulated exocytosis. This insulin travels through the bloodstream and signals other cells to absorb glucose, helping to maintain stable blood sugar levels. When this process is disrupted, when vesicles fail to release insulin properly, it can contribute to conditions such as diabetes. This highlights how essential exocytosis is, not just for communication, but for overall physiological balance. What makes this even more fascinating is the relationship between exocytosis and endocytosis. These two processes are not isolated, they are interconnected and complementary. Endocytosis removes portions of the cell membrane as it forms vesicles to bring substances in, while exocytosis adds to the membrane as vesicles fuse and release their contents. Together, they maintain membrane homeostasis, ensuring that the cell neither shrinks nor expands uncontrollably. It is a continuous cycle of intake, processing, and output, a dynamic balance that keeps the cell stable and functional. When we move from mechanism to real world application, the importance of these processes becomes even clearer. Consider the immune system. When the body is exposed to harmful bacteria, white blood cells respond by engulfing these invaders through phagocytosis, a type of endocytosis. The bacteria are enclosed within vesicles, fused with lysosomes, and broken down by enzymes. This is not just a biological process, it is a defense system, one that protects the body from infection and disease. Without it, even minor injuries could become life threatening. In the brain, exocytosis plays a central role in communication. Neurons rely on the release of neurotransmitters to pass signals from one cell to another. Each thought, each movement, each reaction depends on vesicles fusing with membranes and releasing their chemical messages. Without exocytosis, the nervous system will cease to function entirely. The same principle applies to everyday processes like nutrient uptake. Cells in the digestive system use forms of endocytosis such as pinocytosis and receptor-mediated uptake to absorb essential nutrients, vitamins, and lipids. These substances are too large or too specific to pass through the membrane by simpler means. So the cell actively brings them in, ensuring that it has the resources it needs to survive and function. However, these processes are not always beneficial. Some viruses exploit receptor mediated endocytosis to gain entry into cells. By mimicking normal molecules, they bind to receptors on the cell surface and trick the cell into engulfing them. Once inside, they escape from the vesicle and hijack the cell's machinery, using it to replicate and spread. This reveals a critical insight. While endocytosis is for essential survival, it can also be a point of vulnerability. At every level, from immune defense to brain function, from nutrient absorption to hormone regulation, endocytosis and exocytosis are at work. They are no optional process, they are fundamental to life. They allow cells to interact with the environment, respond to changes, and maintain internal stability. This brings us to some of the most common questions students ask when encountering these concepts. One frequent point of confusion is the difference between endocytosis and diffusion. Diffusion is a passive process. It requires no energy and depends entirely on the movement of molecules from areas of high concentration to low concentration. Endocytosis, on the other hand, is active. It requires ATP and involves complex changes in the cell membrane, including the formation of vesicles. Another common question is why large molecules cannot simply pass through the membrane. The answer lies in the membrane's selective permeability. While small non-polar molecules can pass through easily, larger or charged substances are blocked. Instead of forcing them through, the cell encloses them in vesicles, providing a controlled and efficient means of transport. Students often confuse phagocytosis, and pinocytosis. The distinction is straightforward when viewed clearly. Phagocytosis involves large particles and is often associated with defense, while pinocytosis involves fluids and dissolved substances and occurs more generally. Another question concerns whether exocytosis reduces the size of the cell. Interestingly, it does not. While exocytosis adds membrane to the cell, endocytosis removes it. And the balance between the two ensures that the cell maintains a stable size. Vesicles themselves are not random structures. They originate from specific parts of the cell, such as the Golgi apparatus or the cell membrane, and are formed with precise purpose and direction. Perhaps the most important question of all is whether a cell can survive without these processes. The answer is no. Without endocytosis, the cell will be unable to take in nutrients or defend itself. Without exocytosis, it could not communicate, secrete essential substance, or remove waste. These processes are not just important, they are indispensable. When everything is brought together, the picture becomes clear. Endocytosis brings substances into the cell, exocytosis sends them out. Endocytosis includes phagocytosis for large particles, pinocytosis for fluids, and receptor-mediated uptake for specific molecules. Exocytosis occurs either continuously or in response to signals. Both processes require energy, both involve vesicles, and both are essential for maintaining cellular balance. At its core, the cell is not a passive structure. It is dynamic, responsive, and constantly making decisions. It determines what to take in, what to release, what to keep, and what to discard. Endocytosis allows it to interact with its environment, absorb nutrients, and defend against threats. Exocytosis allows it to communicate, secrete vital substances, and maintain internal stability. Together, they form a continuous circle of intake, processing, and response, a cycle that defines life at the cellular level. And right now, inside your body, trillions of cells are carrying out this cycle, engulfing, packaging, and releasing, working endlessly to keep you alive. If you enjoyed this video, click the like button and also subscribe. Don't forget to also watch out for our next episode that will explicitly explain cell cycle. Thanks.